Good afternoon, everyone. Today I'm before you to present a lecture talk on Mendel's work, okay, then monohybrid cross and dihybrid cross. Okay, now let us go through the list of Mendel's work. Okay, I have listed here some of the Mendel's work. The first Mendel's work was selection of experimental material. Okay, in my previous video I have put all the rest of the information of this, what kind of experiment material he took and what kind of consideration he had while selecting the experimental material. Okay, please go through the previous video of Mendel's work. Second work was Mendel selected 7 pairs of contrasting character for his hybridization or crossing experiment. Okay, hybridization is nothing but cross experiment. He did the crossing experiments with the pea plants. Okay. Third work, Mendel performed several experiments on Pisum sativum. Okay. Fourth work, Mendel did crossing technique. And fifth one is he did reciprocal cross. Then sixth one, pure breeding. Then Mendel cross pollinated 2 varieties of pea plant, that is tall and dwarf. Next work, on the basis of results of his experiment, Mendel recognized the phenomenon of dominance and formulated 2 laws. Okay, then he also worked on monohybrid and dihybrid crosses by explaining 2 laws. Okay, and about these 2 laws he worked and also explained test cross. So these are the some of the Mendel's work. In the exam, if they ask for 5 marks, you can list all these Mendel's work and you can elaborate. Okay, further let us go through now monohybrid. Okay, what is monohybrid cross? Monohybrid cross is the cross between 2 individuals differing in a single pair of contrasting characters. Okay, so let me repeat here. You can give this definition also. A genetic cross between parents that differ in the alleles with one particular gene or single contrasting alleles. Okay, or you can also say here one parent having 2 dominant alleles and other having 2 recessive alleles. You can give this definition for this monohybrid cross, or in simple way you can say it is a cross between the parents, that is individuals, which are differing from single pairs of contrasting characters. Okay, that is known as monohybrid cross. Okay, Mendel in pea plant selected stem length. Okay, in the stem length he selected as follows, that is tallness and dwarfness. The gene for tall is capital TT. The gene for dwarf, that is small tt. Okay, Mendel crossed 2 pure lines. If the genotype, both genotype are same, means that is TT. This is TT, that is the pure or homozygous. 2 pure lines, one tall and another dwarf. Okay, hope you are following me. We are now going through the monohybrid cross. Okay, so for monohybrid cross, he took the stem length, right? In its 7 contrasting characteristics, this is the one of the particular character, that is tall and dwarf. He crossed this tall plant with the dwarf plant. Okay, let's see how he crossed. This is the phenotype. This is the phenotype generation. Okay, whereas here he has taken the homozygous TT and homozygous small tt. Okay, male and female, and parent gametes were single T from the homozygous, again single T from the homozygous recessive. F1 generation, when we crossed, we got capital T and the small t. Okay, this is the F1 gametes. Okay, this is the this is the F1 generation. Okay, next further we can go through F2 generation. Okay, to obtain F2 generation, here we'll be going through self fertilization or self pollination, that is TT is crossed with the TT. We'll get F2 generation. This TT we have written, okay, this is the one generation. Other F1 crossed with F1, another F1 is here. When TT are crossed, we got TT here, capital T, capital T. This T and this T, when we cross capital T and small t here. Small t, capital T. Okay, capital T and small t. This small t and this small t, TT. Okay, this is the offspring. Okay, we got here the phenotypic ratio, that is 3 is to 1. Why 3 is to 1? Capital T tall, capital T small t. It's a tall because T, that is the dominant characteristics. Only dominant characters will express as a phenotypic condition. So this is tall, this is tall, this is tall. So these three are tall. So 3. Then this is the only one which is recessive, that is homozygous small tt. So 1 short. So 3 is to 1. Okay, that is T, tall plant, small t, short plant or dwarf plant. Here also you can go through. Beginning have written here the monohybrid cross definition, and you can also go through this one. Mendel crossed two pure lines, that is one tall and one dwarf. Okay, this is the phenotype and this is the genotype. Capital T, small t, small t. Then you need to take out the gametes. Gametes always you need to put that in the round circle. Okay. Then F1 generation, we got this TT. This is tall. This is a hybrid because one we have dominant allele and another we have this recessive genotype. Okay, then you can write here self fertilization or F2 generation. So to get self fertilization or pollination or to get F2 progeny, we need to cross this F1, cross it with the F1. F1 is crossed with F1, that is capital T, small t is crossed with capital T and small t. This is the genotype. If you want to write here again phenotype, you can write it here phenotype. Tall crossed with tall. That's the phenotype. Then let us take the gametes. Capital T, one gamete, small t, one gamete, cross it with the another gamete, that is capital T, one gamete, small t, one gamete. You can go for this kind of checkerboard or you can also cross like this. This capital T is crossed with the another male or female. If this is the male, this is the female. If this is the female, this is be the male. So this T is crossed with the another T. Then you'll write capital T, capital T. The same T again cross it with the another gamete, capital T and small t. This small t it cross it with the capital T. Then you'll write capital T, small t. Then this T cross it with this T. Then both are small t, small t. So let us write the phenotype. This is pure tall. This is the heterozygous tall. And this is also heterozygous tall. And this is the pure dwarf. So how many tall? 1, 2 and 3. Okay. So when you take a phenotype, tall is to dwarf, 3 is to 1. When you take a genotype here, this is the pure tall. This is the homozygous. Tall is 1 and heterozygous tall is 2. Homozygous dwarf is pure dwarf is 1. So this is the genotype. Okay, 1 is to 2 is to 1. This is the phenotype and this is the genotype. Okay, what explanation you can give here? Here we crossed two pure lines, that is tall and dwarf. Okay, when we got the F1 individuals, we got here the heterozygous, right? Capital T and small t. In F2, what happens here? Two types of individuals we got here. That is 3 by 4th or 1 by 4th. 3 by 4th tall and 1 by 4th dwarf. This showed him that characters for dwarfness was disappeared in the F1 generation, which was reappeared in the F2 generation. Okay, so this is all about the monohybrid. If you want, you can take screenshot of this particular slide and you can make a note. Hope you understood about monohybrid cross. Okay, now let us go through what all you need to keep in your mind while writing this particular cross. First, you need to write phenotype, okay, then genotype, then gametes, then F1 generation, you'll get after F1 generation, self-pollination or self-fertilization, then you'll get the F2 generation cross. Then later, after the cross, you need to write phenotypic ratio is what and genotypic ratio is what, okay. So this is all about the monohybrid cross. Now let us go through The explanation. Okay, you already understood this one. Mendel crossed two pure lines, that is tall and dwarf, where F1 individuals with a tall hybrid, he is heterozygous, right? He then selfed with F1 heterozygous, that is tall crossed with the tall. Here he got in F2 obtained two types of individuals, that is 3 by 4th of tall plants and 1 by 4th of dwarf plant, that is in 3 is to 1 ratio. Okay. 3 is to 1 ratio is the phenotypic ratio, whereas 1 is to 2 is to 1 ratio is the genotypic ratio. This showed him that the character for dwarfness disappeared in F1 generation, which was reappeared in the F2. Okay. So we can give the definition of the monohybrid. That is, monohybrid is the cross between the two individuals differing in a single pair of contrasting character. Okay, this is all about the monohybrid. Now let us go through the phenomenon of dominance. The character of dwarfness was not expressed in F1, even though the individuals of F1 carried the genes for dwarfness, right? This is because the tallness is dominant over dwarfness. Thus, the character which expresses itself in F1 hybrids are called dominant character, while that which is not expressed or suppressed in an F1 hybrids are called recessive character. Okay, kindly go through this sentence again. You need to keep in your mind which one is dominant character and which one is recessive character. Okay. Now let us go through dihybrid cross. How could you define dihybrid cross? Here, the hybridization of two individuals which differ in two pairs of contrasting character known as dihybrid cross. Okay. In pea plant, Mendel selected seed form and seed color. Okay. Here you can see two characteristics. Monohybrid only. The stem length. Here there are two characteristics. What are those? One is the seed form. Other one is the seed color. Two contrasting characters of an individual. Okay. Both of them are having a pair of contrasting character as follows. In seed form, if you are taking here the seed form means in the seed form, if you are taking the seed form, here the form is round and wrinkled. Seed color is yellow and green. These two are the contrasting characteristics. Okay. Now let us go through that which characteristics we have taken. Seed shape character, smooth or wrinkled or round or wrinkled. Seed color, yellow or green. Okay. This is the dominant characteristics and this is the recessive character. Now we can go through the cross. Okay. Gene for round and yellow represented as capital RR and capital YY. Always dominant character will be represented in the form of capital letters and recessive characters will be in the small letter. Okay. Gene for wrinkled and green represented as small RR and small YY. Okay. Kindly follow me and if possible write in your notebook. Mendel crossed pure lines of round yellow and wrinkled green seeds. Let us write the cross first, that is phenotype. Okay. Phenotype, next genotype, then gamete. Okay. First parent phenotype round yellow crossed with wrinkled green. Then genotype for round, it's capital RR, yellow capital YY. Okay. For wrinkled green, small RR and small Y. Then let us write the gamete. Well, gamete, homozygous capital R or 1R, homozygous capital Y, 1Y. Okay, homozygous small r, one single R gamete, and then homozygous small y, 1Y. So now gametes are ready. Okay, once the gametes are ready, then you can get the F1 generation. In F1 generation, we'll get this F1 generation, that is the hybrid RRYY. So this is the hybrid we got, which is the round yellow character, that is heterozygous. Okay. Next generation will go through self-fertilization or self-pollination. This F1 is crossed with F1. We got RRYY. This RRYY is crossed with RRYY. It means here one will be considered as male, other will be as female. So here let us take the gametes. This R is crossed with this capital Y. The same R again with this small y. This small r crossed with the y, capital Y. Then this small r crossed with this y. So we got here the four gametes from the male and the same gametes from the female. Now once the gametes are ready, you can plot this checker board. Okay, so male and female. This ry is written here. This ry is written here. Small r capital Y is written, small y is written. Same all these gametes which are written here, one side male, other side female. Now we'll cross here. Okay, this R and this R, Y, Y, R, R, Y, Y. Okay, this R, capital Y and small y, R, R, Y, Y. Like this you'll cross and ultimately you'll be able to get all this green color, what a triangle box have put here. No, this is round yellow. Okay, this is round yellow. And next one you will get here, this is red color one is the round green. Okay, and the next one is wrinkled yellow. Okay, this is the wrinkled yellow. And next you will get here that the last one is wrinkled green. Okay, when you take all this particular ratio phenotypic ratio, that is round yellow, this one, then round green, this red one, then wrinkled yellow, this green one, and the blue one wrinkled green, which comes under 9 is to 3 is to 3 is to 1 ratio. And this is the genotypic ratio, okay, which gets in 16 combinations. Okay, that is 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. So total 16 combination. So this is all about the dihybrid cross. If you want, you can pause this video and you can make a note. Further, let us go through here the explanation about this dihybrid cross. The gene for round and yellow represented as capital RR, capital YY. The gene for wrinkled and green represented as small rr and small yy. Mendel crossed pure line, that is round yellow and wrinkled green seeds. Why I'm giving this after the cross, below you need to write the explanation. Okay, in F1 generation, all the plants produced are round yellow seeds, but they were heterozygous. Okay, parent phenotype, they were actually homozygous, but we got in F1 generation heterozygous. When the F1 hybrids were allowed to self-fertilize, in F2 generation, what has happened there? We got 16 different combinations, right? In the ratio of 9 is to 3 is to 3 is 9 is to 3 is to 3 is to 1 ratio, that is round yellow, round green, wrinkled yellow, and wrinkled green, right? Among these four types, round yellow, wrinkled green are the parental phenotype, right? They resemble parent characteristics, while the yellow, wrinkled, and round green were recombinants, right? The cross can be explained with the help of checkerboard or Punnet's square. Okay, this is the explanation you're supposed to give below the dihybrid cross. Next, you can keep in your mind this diagrammatic representation of cross also. This is the parent generation plant grown from round yellow pea plant, then plant grown from wrinkled green pea. Okay, then we got the gametes here. F1 generation, all seeds produce round and yellow with heterozygous condition. Then this will go further, move to the self-fertilization in F1 plants. Okay, after self-fertilization in F2, you'll be able to find 4 different pea phenotypes, right? That is round yellow, then round then wrinkled yellow. The next one is round green, then wrinkled green. Okay, that is in the ratio of 9 is to 3 is to 3 is to 1. Okay, this is the checkerboard. You can go through. You can remember this one or the previous one also. You can go through. Okay, I have written here phenotypic ratio 9 is to 3 is to 3 is to 1. Okay, then genotypic ratio I've written. Okay, I think here there are Uh, 16 combinations are there, I think. Please check. 1, 2, 2, 4, 1, 4, 1, 2, 1, 2, 1. There are some mistake here. Okay, now it's correct. Okay, this is the 16 combinations of the round yellow and wrinkled green, then wrinkled yellow and brown green ratios. Okay, you can also keep this in your mind. Okay, these are the different ratios. I hope you understood today's Mendel's cross. Okay, thank you everyone for watching this video.